Hello students, I am Dr. Nilesh Sawan and I am back today with the last lecture on the topic respiration. So we have complete with the entire respiration part. So some few topics associated with respiration are left. So we'll cover up those things. So in the last lecture what we saw was the ETS. So we'll first see immediately the significance of ETS. Now if you observe it clearly, in glycolysis, if we see, total actually 6 ATPs are derived from NADH2. NADH2 will give you 6 ATPs and 2 ATPs are direct ATPs. Means actually 4 ATPs are direct ATPs, but 2 ATPs we have used in the process. So 4 minus 2 will give you 2. So out of that 8 ATPs, these ATPs are released via ETS. Means this NADH2 has to enter the ETS cycle to give those ATPs. Simultaneously, in Krebs cycle, there are 8 NADH2 formed. 2 of those are from the linking step. Then we have 2 FADH2s. So, and 2 substrate level ATPs, that is in the form of GTP, they are formed. So we'll take it directly as ATP. So here 8 3s are 24 ATPs and this is 2 2s are 4 ATPs. So in total these 28 and these 6. So in total actually 34 ATPs out of 38 ATPs first of all are generated by ETS. These are first generated by ETS cycle. So that's a very important part. So out of the total 38, we will get those 38 ATPs when they enter into the ETS, electron transport chain. So this is how it goes. So glycolysis ke 6 ATPs and 28 ATPs of Krebs cycle will be formed actually in ETS, not in glycolysis, not in Krebs cycle. So that's the first advantage. Then it will regenerate back. It can regenerate NAD plus and FAD plus. So these hydrogen acceptors, इलेक्ट्रॉन एक्सेप्टर्स दोनों हम बोल सकते हैं. Generally these are again regenerated back because NADH2 will break down, मतलब it will liberate its hydrogen and it will again form NAD plus. ठीक है, NAD plus in that sense. Next one, importance of ETS is what it will help in formation of water. ठीक है, help in formation of water which is needed in Krebs cycle. So this will be released in the matrix. So the byproduct formed, that is water, is released into the matrix of the mitochondria. So that provides it the additional what you can say, the required solvent that is needed. ठीक है, and significance में ये 1 2 3 and I think so one more point which they have given is formation of water. And yes, so एक stepwise इलेक्ट्रॉन ट्रांसपोर्ट में एनर्जी रिलीज होती है. इट इज नॉट रिलीज्ड इन व्हाट यू कैन से इन अ बल्क, सो इट हेल्प्स टू प्रिवेंट द डैमेज ऑफ सेल्स. सो दैट इज वन ऑफ द इंपॉर्टेंट पॉइंट्स. सो दीस आर द 4 सिग्निफिकेंस ऑफ ETS. होप सो यू हैव गॉट दिस. सो ETS के 4 सिग्निफिकेंस लर्न करो. 2 मार्क के लिए कॉलेज एग्जाम्स में आ सकते हैं. 11th स्टैंडर्ड है तो फिर कॉलेज एग्जाम्स में पूछ सकते हैं. नाउ व्हाट इज एम्फिबोलिक पाथवे? नाउ Krebs cycle is called as actually amphibolic pathway. Now normally what we see is nothing but a catabolic. Catabolic means generally there is breakdown, as we all know. In Krebs cycle acetyl-CoA oxaloacetic acid will first form citric acid, then it has been broken down from 6 carbon to 4 carbon. So normally what we see is it's a catabolic reaction. So but instead the byproducts, the substituents which are formed like acetyl-CoA, alpha-ketogluteric acid, then oxalosuccinic acid. Actually, these chemicals which are actually formed in the Krebs cycle can also be used in formation of, matlab anabolically they can be used to make amino acids, alpha amino acids. They can be used to make fatty acids. So technically, from these substituents which are formed in the Krebs cycle, we can synthesize new products also. So technically speaking, if we just see Krebs cycle, it's a catabolic process, but these compounds can also be used to prepare amino acids and all that stuff. So ये anabolic भी है. So Krebs cycle technically is anabolic and catabolic both. That's why we are calling it as amphibolic. So it shows reactions. Now obviously for respiration we will only see the catabolic cycle, but practically in plants, जैसे mitochondria, we also know they have ribosomes. So they might require certain amino acids to prepare protein, and by chance if those amino acids are not available, then these substrates of Krebs cycle are utilized to prepare them, and when needed they have been broken down, or rather they have been used to broken down, and they will be used to form back these substituents. So these reactions take place in mitochondria. So the degradation takes place, but from this formation of useful substance also takes place. When necessary, these useful substances are again broken down into these so as to run the Krebs cycle. So in this way you can see it is showing anabolism and catabolism both. That's why we call this Krebs cycle as amphibolic pathway. And इसका एक फायदा है, तो इसी से ये एक इम्पोर्टेंट फीचर है कि why stepwise energy is released. Stepwise energy release, utilization of energy stepwise होती है. क्यों? इसके फायदे क्या है? What are the, मतलब सिंपल, when we see suppose if you burn a wood, लकड़ी अगर हम जलाते हैं तो ऑब्वियसली उसमें भी से तो एनर्जी निकल ही रही है. यहाँ पे भी रेस्पिरेशन में भी एनर्जी निकल ही रही है बाय बर्निंग दी सबस्ट्रेट, बट ये बर्निंग वो बर्निंग से अलग है. मतलब लकड़ी जलती है तो देखो उसके ऊपर कोई कंट्रोल नहीं होता अपना. मतलब when we burn a wood, the fire and the reaction which takes place inside it cannot be controlled by us. मतलब वो जोर से, मतलब अगर कभी स्टार्टिंग में थोड़ा आग स्लोली जलेगी, फिर शायद भड़क उठे एंड देन इट कैन गिव यू अ लार्ज फायर. बट ये हम लोग अफोर्ड नहीं कर सकते व्हेन वी ब्रेक डाउन ग्लूकोज. ग्लूकोज से अगर एक झटके में सारी एनर्जी निकल गई, देन इट विल बी डैमेजिंग फॉर सेल. सो सबसे पहला तो वही है, स्टेप वाइज रिलीज ऑफ एनर्जी. फर्स्ट, it allows full utilization of substrate, and release maximum ATPs, max ATPs. So तुम देख सकते हो एनारोबिक में भी किया था तो 2 ATP मिले, बट जब हम स्टेप वाइज मेथोडिकली जब उसे ब्रेक डाउन करते हैं तो उसी ग्लूकोज से 38 ATPs भी निकाले जा सकते हैं. अगर हमने जैसे एब्रप्टली अगर ब्रेक डाउन किया तो उससे 2 ही ATP आते जो हमने अल्कोहलिक फर्मेंटेशन, लैक्टिक एसिड फर्मेंटेशन में देखा. तो इन स्टेप वाइज तरीके से ब्रेक डाउन करने का फायदा ये है. दूसरा, each reaction is enzyme control. We know that each reaction of glycolysis, Krebs cycle and even ETS is enzyme control. So we can easily say accelerate or inhibit certain reactions, and we can means the cell can control this respiration, means the rate of respiration can be controlled. See, normally when we are sleeping, we breathe slowly and we respire, and that time energy release is also in a lesser amount. But when we do some strenuous exercise, now that time body needs more energy to compensate that. So that time these enzymes will act proactively and they will bring about this conversion very fast. So each reaction is enzyme control, hence respiration can be accelerated, means whenever the body needs it, it is accelerated, or when not in need that much, it can be inhibited or it can be slowed down. So there is an effective control of the cell because of the enzymes, because the release of enzymes is under the control of the cell, because the formation of enzyme requires nucleus, and nucleus indirectly will will take the call. And last but not the least, एक फायदा ये है amphibolic pathway. Intermediate substances that are formed that can be a part of two different cycles or two different chemical reactions. Now जैसे मैंने बोला अल्फा कीटोग्लूटरिक एसिड वगैरह ये रेस्पिरेशन के सबस्ट्रेट्स है, बट इफ जैसे एग्जांपल होता है व्हेन वी आर स्लीपिंग, दैट टाइम द रेट ऑफ रेस्पिरेशन इज नॉट फास्ट, सो ड्यूरिंग दैट टाइम जो ये इंटरमीडिएट सब्सटेंस है उनकी उतनी जरूरत नहीं होती है शरीर को, बट it can be used to make some different useful things like amino acids, fatty acids. Normally again when we wake up or so, when we do some exercise and all, that time we require more of this respiratory substrate. So those amino acids, those fatty acids have been broken down to get you to this Krebs cycle. So the substrates of Krebs cycle. So that's a what you can say an advantage of stepwise energy release. First, effectively that substrate is broken down and what you can say maximum energy can be derived from that substance. That's the first advantage. Dusra, since they are enzyme controlled, we can accelerate it and we can even slow down it. Inhibit would not be a great word, I think so. Accelerate or slow down would be the better option. Okay, slow down or accelerate. Inhibit means to stop directly, so that is not going to happen, but obviously only after death that is possible. And last, intermediate substrates. So in this way the substrates can be reused somewhere and again be formed again for Krebs cycle. So that's the advantage of utility of the stepwise energy release during the entire respiration. Now the next one is some experiments they have given. So experiments देखो क्या है, तो उन्होंने क्या किया, just to prove anaerobic respiration. दोनों experiments is there to prove anaerobic respiration. Now this is say test tube 1, which is filled with 10% solution, of glucose. सादी भाषा में पानी और ग्लूकोज का मिक्सचर है जिसमें ग्लूकोज का कॉन्सेंट्रेशन 10% दिया है, ठीक है, ऐसा याद रखो. Now what they have done, they have added Baker's yeast. Means yeast we all know it's a unicellular fungus, but yeast की एक खूबी है, it can whenever oxygen is available it will show aerobic, but when oxygen is not available it shifts to anaerobic mode of respiration. Next what we are going to do is we are going to add little bit of oil over here, means slowly and carefully. So what this will ensure कि no oxygen is entering into this, ठीक है, no oxygen is entering into this, that we have ensured over here. Then what we are going to do is we will fit a tight rubber cork so that air cannot enter or go out. So here only there will be a little bit of air. Then we will pass a glass tube through this, which will be opening in this open area, ठीक है, ये open area जो है वहां पे ये खुलेगा. Next यही हम कनेक्ट करेंगे एक दूसरे टेस्ट ट्यूब को we are going to connect this, and in this test tube it consists of lime water. What is lime water? It is CaO, ठीक है. And then we'll connect, this is also tightly fitted with a rubber cork, and this is connected to this one, and this is dipped into this. So make sure this tube is dipped inside the test tube B. So this is test tube A, okay, which is filled with water, and in that 10% glucose is added, and also yeast, okay, which is a unicellular fungus which is added over there, so that glucose serves as a food for it. Then we have sealed it with a layer of oil, because oil will ensure no air goes into this solution. Then we have tightly fitted this rubber stopper, and through that we have passed a tube which connects to the test tube B, and it is immersed inside the solution of lime water. So this is lime water to detect the presence of carbon dioxide, and this is the setup. Now we'll put this in a warm setup, test tube A this is. So we'll put it in a warmer or in an area where the temperature is 35, 36, 37 degrees. Why? Because this will stimulate or trigger the yeast to start working. Enzymes will start to work. Now initially what will happen is, you can see it will go under anaerobic mode of respiration. So what it is going to release here, it will be full with CO2, because in this it will undergo alcoholic fermentation. So a byproduct is formed, that is CO2. So this CO2 is going to build up, and then since it has no place to go, so it will come out and it will be present over here, and from here it will travel through this, and here this CO2 will combine with lime water, and hence this lime water will turn milky. So if the lime water is turning milky, this is a clear-cut indication that the yeast has released CO2, because CO2 only will turn lime water milky. Now let it be there for 2 to 3 days, okay? And later on remove this stopper and just smell it. You will smell like alcohol, because that glucose will be fermented to alcohol. So the presence of CO2, which can be easily seen when lime water turns milky, the presence of, or rather formation of ethanol, which is formed in the test tube A, this confirms that there was anaerobic respiration which took place, fermentation you can call it, was done, and this is how yeast has performed anaerobic respiration. So this is a confirmation that yeast has performed anaerobic respiration, because we have got alcohol as well as CO2 as a products and byproducts. So this confirms the yeast performs anaerobic respiration in absence of air. In presence of air, it will also do aerobic respiration only. So first experiment, hope so सबको समझा होगा how it works. Let's see the second experiment. Now second experiment is also same, to prove anaerobic respiration takes place in germinating seeds. So what they have seen is, initially what they have done is, they have taken a test tube and filled it with few germinating seeds whose seed coats have been removed. Seed coats remove होने से germination आसान हो जाएगा. So there are few seeds taken over here, and then they were poured with mercury. This was filled with mercury. Now obviously mercury is heavy, so it will also be covering this also. Okay, now what we are going to do is, we will take a tub filled with mercury. We will place our thumb over here. On this to close this. Okay, so if suppose I'll take a chalk to do this. Suppose this is a test tube, so I have put my thumb over here. There are seeds and mercury over here. Now what I will do is, I will take this test tube and I will immerse my thumb inside the mercury and I will leave it. Now what will happen is, since seeds are lighter, okay, they will go up and the mercury will be over here. So we will dip till here, till entire the air pocket over there is gone. Means what we will be doing is, we will put this test tube inside such that the mercury fills the entire test tube, and here what we are left is with the seeds. Okay, now you can just lift it little bit over here. So we'll leave this inside the tub. Now what will happen, since there is no oxygen, no air over there, they will go under, they will show anaerobic respiration. How it is proved is, they will produce CO2, and anaerobic respiration, if they will show, so they will produce what? CO2. Now gas occupies, every matter occupies space, So obviously that CO2 will push this mercury little bit downwards. Okay, so that CO2 gas is formed and it will make a or rather it will develop a pressure which will push the mercury down. Obviously little bit level of mercury will increase over here. Now if we put a piece of potassium hydroxide over there. Now potassium hydroxide has a capacity to absorb CO2. Now this has a capacity to absorb CO2. So this pellet is also light. So what we'll do, we'll put this pellet over here and we'll leave it. This will rise and reach at the top because of its lighter weight. Now the entire CO2 which was formed will be absorbed by this, and this will get converted into potassium carbonate. Obviously since the gas has been removed, the mercury level again rises over there, and obviously this is what we again get. So this proves that though the seeds have released CO2, that means they are showing anaerobic respiration. So the presence of CO2, and how we have guaranteed that? So when we put KOH, it has been converted into K2CO3 and water obviously, and that will result into formation of back pressure, means the gas, since it is removed, it will create a vacuum which will again pull the mercury level up. So this shows the seeds have produced CO2, and that's how we have to show that. So these two experiments, they just tell you in absence of air, anaerobic respiration can occur. So that's the only bottom line that you need to understand. Next one is respiratory quotient. Now what is respiratory quotient? Respiratory quotient is amount of CO2 released, amount of CO2 released upon amount of oxygen used, amount of CO2 released upon amount of oxygen used. So एखादे substrate को degrade करेंगे, because we know respiration is a catabolic process. So the complex substrate will be broken down into CO2 and water. We all know this. But to do that we require oxygen. We know this. So simple है. अभी एक एग्जांपल ले रहा हूँ. 3/2 की वैल्यू बड़ी आएगी कि 2/3 की वैल्यू. So obviously this will be 1.5. This will be 0.6 roughly. ठीक है. So 6/18, 3/6/18. So roughly ये वैल्यू आएगी 6666 आता जाएगा. So तुम देख रहे हो, when numerator is large, value is higher than 0. When denominator is large, the value is below 0. Here you can see this. Now similarly, obviously if CO2 release is more than oxygen used, the value will be higher. हमें यही चाहिए कि कम ऑक्सीजन में कौन इजीली ब्रेक डाउन होता है, एंड दिस इज व्हाट आवर सेल डिसाइड्स. मतलब सबसे बेस्ट सबस्ट्रेट कौन सा होगा सेल के लिए, तो उसके लिए ये वैल्यू निकाली जाती है. मींस सी वी नो वी हैव 3 बायोमोलिक्यूल्स. कार्बोहाइड्रेट्स, मतलब अपना शरीर रेस्पिरेशन के लिए इन्हीं का इस्तेमाल करता है टू डिराइव एनर्जी. एक तो कार्बोहाइड्रेट्स, एक तो प्रोटीन्स, एक तो फैट्स. एक और बॉडी यूज कर सकती है बट वी रेयरली यूज दैट इज ऑर्गेनिक एसिड्स. ये कुछ दूसरे एनिमल्स में देखने को मिलता है. नाउ if we calculate the what you can say respiratory quotient of these substrate, the value of carbohydrates is 1, the value of protein is 0.9, fats is 0.7, and organic acids is greater than 1 surely. Some have 3, some have 10, depends कि organic acid कैसा है. So first of all ये तो हम rarely use करते हैं. This is used by some plants. ठीक है, in some plants these organic acids are broken down to get energy. So we'll debate on this. So you can see out of carbohydrates, proteins, fats, highest value किसकी आ रही है? Carbohydrates. इसलिए सेल्स कार्बोहाइड्रेट्स चूज करते हैं फॉर प्रोड्यूसिंग एनर्जी, बिकॉज जितना ऑक्सीजन यूज होगा उतना ही CO2 रिलीज होगा. इसका मतलब दिस रेशियो विल कम एज 1. बट लुक ओवर हियर, द वैल्यू इज लेस कब होगी? ज्यादा ऑक्सीजन होगा तब CO2 निकलता है. मतलब to release CO2 from by degrading protein and fats, we have to take in more oxygen. So तुम समझ सकते हो सेल्स के लिए घाटे का सौदा है, मतलब टू बर्न फैट्स एंड प्रोटीन्स बॉडी रिक्वायर्स मोर ऑक्सीजन एज कंपेयर टू कार्बोहाइड्रेट्स, इसलिए बॉडी प्रिफर्स टू बर्न कार्बोहाइड्रेट्स. अच्छा चलो कार्बोहाइड्रेट्स खत्म हो गए या कम हो गए, नेक्स्ट प्रिफर्ड एक्चुअली प्रोटीन्स है बॉडी के लिए. बट बॉडी ये भी जानती है प्रोटीन्स आर वेरी मच एसेंशियल मॉलिक्यूल्स बिकॉज दे आर नीडेड फॉर बिल्डिंग ब्लॉक्स ऑफ सेल्स, फॉर एंजाइम्स, हार्मोन्स, एंटीबॉडीज एंड मेनी मोर थिंग्स टू गो. इसलिए देन बॉडी गोज विद फैट्स. नाउ हियर यू कैन सी, मतलब ये थोड़ा मैं बताना चाहूंगा जो लोग एक्सरसाइज ओरिएंटेड है, मतलब जो ओबेस है और उन्हें फैट बर्न करना है, यू हैव टू यूज लॉट्स ऑफ ऑक्सीजन. एंड लॉट्स ऑफ ऑक्सीजन कब यूज होता है, तो व्हेन यू डू वेरी स्ट्रेनस एक्सरसाइज. इसलिए फैट्स बर्न करने के लिए यू नीड इंटेंस एक्सरसाइजेस जिसमें बॉडी हैज टू गैस्प फॉर ऑक्सीजन एंड दैट टाइम योर बॉडी विल बर्न द फैट्स. ठीक है, सिर्फ प्लेन वॉकिंग इज नॉट, मतलब उसके लिए फिर तुम्हें बहुत चलना पड़ेगा विच इज नॉट फीजिबल. ठीक है, बट इफ यू डू, जिसे अपन कहते हैं हाई इंटेंसिटी इंटरवल ट्रेनिंग, कि 15-20 मिनट्स में यू गो योर heart out and do those exercises, heavy compound movements, this will help to boost your metabolism because body then use lots of energy. That time we also know carbohydrates के मुकाबले fats ज़्यादा energy देते हैं. 1 gram of fat will give you 9 kilocalories, proteins will give you 4, carbohydrates will give you 4 kilocalories 1 gram. So body also knows कि fat has more potential to give energy. So during very high strenuous exercises, body then will burn fat. But before that, body has to first deplete all the carbohydrates in the body. So carbohydrates खत्म हो जाए, इसलिए normally हम बोलते हैं the person who wants to lose fat should train or should go for exercise empty stomach. Because say suppose example दे रहा हूँ, रात को खाना खाएं, early morning if you go for gym, or that sort of, if you go for cycling, if you go for running, any of these can do. That time your body is depleted because for after 6 to 7 hours of sleep, no doubt body has used up lots of carbohydrates. So some of the carbohydrates which will be burned in initial half of your exercise, and later on body will then burn fat. Okay, but don't directly compensate eating a huge amount of meal after that, because हमें ये maintain रखना है. This is known as calorie deficit. एंड इसलिए बॉडी ज्यादातर कार्बोहाइड्रेट्स इजी पड़ते हैं टू बर्न, इसलिए बॉडी ऑलवेज प्रिफर्स दिस. सो रेस्पिरेटरी क्वेश्चन हेल्प्स अस टू अंडरस्टैंड व्हिच सबस्ट्रेट इज बेटर फॉर रेस्पिरेटरी पर्पस. दैट इज ऑब्वियसली द कार्बोहाइड्रेट, दे आर द सिंपलेस्ट वन. नेक्स्ट एक्चुअली आर प्रोटीन्स बट बॉडी प्रिफर्स टू बर्न फैट्स देन प्रोटीन्स. ठीक है, सो दीज आर सम ऑफ द रेस्पिरेटरी क्वेश्चंस रिलेटेड टू रेस्पिरेटरी क्वेश्चन. And the last one is significance. What's the importance of respiration? So first तो मैंने बताया it provides the biological energy that we require to do all our activities. Secondly, first though it's a source of energy. Second, it breaks down the complex compounds into simple inorganic ones. Third, regarding respiration is third one, it's a process which is in tandem with photosynthesis. So it helps to keep the balance of what you can say the oxygen and CO2 level in the environment. ठीक है, so ये significance of respiration, again stepwise breakdown of glucose molecules वो भी दिया है, plus some new substrate like amphibolic pathway भी मैंने बताया था. So substitutes of respiration are also used for synthesis of other substances. So that also plays a very key role. Then anaerobic respiration. So पूरी wine industry, bakery industry, they rely on that fermentation part. So the knowledge of this respiration will help us to develop good quality wines, then bakery products and all that stuff. So this is the significance of respiration. Even respiration, what you can say, with anaerobic respiration we can say lactic acid, because lactic acid fermentation which takes place in our body, it helps the body to keep working continuously. Because there's an example, if you if you are exhausted but you still want to do some work, then body has to go in that phase. Okay, so in case of strenuous work, body can switch over to anaerobic respiration. I know that is not a very swift way because the energy output from anaerobic is less, but still it's better than nothing. ठीक है, if oxygen demand, if the intake of oxygen is somehow reduced, then body can switch over to that also. So that is one also important aspect of respiration. तो होप सो गाइस ये रेस्पिरेशन टॉपिक सबको समझा है. सिग्निफिकेंस तुम बुक से भी पढ़ सकते हो, इजी टू अंडरस्टैंड है. मैंने मेन सिग्निफिकेंस तुम्हें बताया है. सो गाइस मिलते हैं नेक्स्ट वीडियो में, जिसमें हम लोग अभी शुरू करेंगे एनिमल के प्रोसेसेस. सो वी हैव टू लुक फॉर डाइजेशन, ह्यूमन स्केलेटन एंड ऑल दैट स्टफ. सो एक्सक्रीशन आल्सो. सो लेट्स मीट इन द नेक्स्ट video or the next lecture. Till then guys, hit the like button, subscribe the channel. See you later.